now turn our attention to DNA. Genes are discrete hereditary units located on the chromosomes. Each gene provides a clear and unambiguous set of instructions for producing some property of its organism. As you should have noticed on the ideograms of the X and Y chromosome, each chromosome includes hundreds to thousands of genes. The complete set of genes in an organism is referred to as its genome. Genes are made of deoxyribonucleic acid, or DNA. A DNA molecule consists of two long strands wound tightly around each other in a spiral structure known as a double helix. The structure has been likened to a twisted ladder whose sides are made of sugar and phosphate and whose rungs are made of bases. Each strand of the DNA molecule, that is, each side of the ladder once it has been untwisted and straightened out, is a linear arrangement of repeating similar units called nucleotides. Nucleotides are interconnected by the linkage of the sugar-phosphate groups, forming photodiester bonds between them on a single strand. The basic unit, or nucleotide, is composed of an organic base attached to a deoxyribose sugar, and the phosphate is also attached to the sugar. The base is one of cytosine, which we refer to as C, thymine, which we refer to as T, adenine, which we refer to as A, and guanine, which we refer to as G. This is the chemical structure of DNA, which is a double helix with bases held together like rungs on a ladder and sugar phosphate units along the sides of the helix, so it forms a sort of twisted ladder. The chemical structure of the four bases, A, T, C, and G, is shown. Each base is then attached to a deoxyribose sugar, and a phosphate group is attached to the sugar to form the sugar-phosphate backbone. Many years ago, Chargaff's rule determined that all cells have the same number of A's as the number of T's, and the same number of C's as the number of G's. And in fact, this is because the two sides of the ladder are complementary. An A base pairs with T, while a C base pairs with G. This is known as Watson-Crick base pairing rules. These pairs are held together by hydrogen bonds. Interestingly, there are 2 hydrogen bonds between A and T, while there are 3 hydrogen bonds between C and G. The force that holds a base pair together is a weak hydrogen bond. Although each individual bond is weak, their cumulative effect along the strands is strong enough to bind the 2 strands together tightly. As a result, DNA is chemically inert and is a stable carrier of genetic information. This is a picture of James Watson and Francis Crick, who were 25 and 37, so important discoveries can be made while you were young. Of course, their structure was presumed because of work by others, including Rosalind Franklin, who obtained images of DNA using X-ray crystallography. An important concept is the central dogma of molecular biology. DNA is the molecule that carries inherited information. Duplication, most commonly called replication, is the biological process of producing 2 identical replicas of DNA from one original DNA molecule. In this way, our cells pass genetic information through generations of cells. Transcription is the making of a single-stranded RNA molecule off a DNA template. There are many types of RNAs, including messenger RNA, transfer RNA, ribosomal RNA, microRNA, long non-coding RNA, etc. Translation is the construction of an amino acid sequence into a protein from an RNA molecule. Proteins are the molecules that carry out most of the biological functions. As an aside, there are certain RNAs that have special functions, such as they can reproduce themselves, like the coronavirus. Also, some RNAs can be reverse transcribed or turned into DNA, like the HIV virus. Note, though, that this is rare. Next, please watch the YouTube video illustrating DNA replication.